So the next speaker is Dr. Quentin Raas. He's a postdoctoral fellow in Dr. Bonkowski's laboratory at the University of Utah, and he will talk about the zebrafish, I think, right?
Okay, good morning, everyone. I want to start by thanking the organizers for the great opportunity to talk today. So my name is Quentin Raas. I'm a postdoctoral fellow in Josh Bonkowski lab at the University of Utah. And so yes, I will be talking about the zebrafish model of XALD. So very brief repeat of what has been said about XALD. Sorry. So it is associated with mutations. Oh wait, it doesn't show it. Okay. So XALD is associated with mutations in ABCD1. Its incidence is believed to be around 1 for 15,000 births, and the disease can manifest with very distinct forms. It's known to affect the adrenal glands and to cause Addison disease in about 80% of the patients. But the most severe form of the disease is probably this demyelinating form that is affecting the white matter in the brain. And it's often referred to as cerebral ALD, or also childhood cerebral ALD, as it's often affecting patients at a very young age, before the age of 10. And it's estimated to affect about 50 to 60% of the patients if we consider the combined incidence in children and adults. And finally, the neuropathy, representing the adult form of the disease, called adrenomyeloneuropathy, which is affecting the spinal cord and peripheral nerves, and is believed to touch most patients. So the ABCD1 gene is encoding a transporter that is found at the membrane of peroxisomes, which imports very long-chain fatty acids into the lumen of peroxisomes, which is the exclusive site for the degradation by beta oxidation. And so this is its deficiency is causing very long-chain fatty acids to accumulate in different tissues, and this represents the main biomarker of the disease as well. So this is certainly the most discussed point, as VLCFA accumulation is believed to affect the myelin sheath and potentially leading to demyelination, but there are no clear evidence for this. And there is also no correlation between the severity of the disease and the accumulation of very long-chain fatty acids. So there is a crucial need for experimental models with all these questions remaining, and obviously also to lead to efficient therapies for both adrenomyeloneuropathy and for cerebral ALD. As some of you know, also there are currently limitations with animal models, as for example the ABCD1 deficient mice only develop a mild phenotype starting at a very advanced age after about 15 months. And so that's why we think zebrafish can be an interesting alternative here. Just a few points about zebrafish. So they are the small fish which you can house for only a fraction of the cost of stabulation for mice, and so housing easily hundreds or thousands of individuals. And what's definitely interesting is that a single cross of one male to one female can yield several hundred embryos, as you can see in this plate, and that just make that model a very good model for large-scale screens. And so in our lab we have developed some tools, and we have device to just generate large automated screens with, for example, recording of the behavior of the zebrafish larvae in a 96 well plate format. The zebrafish have also a rapid and transparent embryonic development, with myelination starting only about 2 days post-fertilization. And after 4 to 5 days, the fish is already showing a robust myelination in most of the central nervous system. And similarly, the blood-brain barrier starts forming between 72 and 96 hours. So after 4 to 5 days, we already have a model organism that is showing interesting properties in regards to the study of leukodystrophies and things like neuroinflammation. We have also many molecular biology tools enabling rapid and efficient generations of transgenic zebrafish for the expression of fluorescent reporters in specific tissues, such as in endothelial cells and also in microglia or macrophages, as here. And in that regard, we also generated transgenic fish expressing a red fluorescent protein with this conserved peroxisomal targeting signal, similarly that what has been done in Cholan's lab. And we can here visualize peroxisomes in the spinal cord in motor neurons, for example. And we generated also different tissue-specific lines with here peroxisomes in macrophages. And quite interestingly, we have these very distinct tissue-specific dynamics of peroxisomes with macrophages exhibiting those very rapid movements of peroxisomes. And so I believe these can make good models to study peroxisome dynamics and interactions. Finally, when considering the gene conservation in zebrafish, it's important to note the strong conservation of the vast majority of genes in vertebrates, for more than 70% of it. And interestingly, peroxisome genes and metabolism is very well conserved, and especially beta oxidation. And to illustrate this, when comparing the amino acid sequence of ABCD1 in human and in zebrafish, we measured about 70% of amino acid identity, and we can also tell that the gene is expressed in the central nervous system, as shown here by in situ hybridization, and it's expressed in colocalized pretty well in oligodendrocytes precursor as early as 48 hours post-fertilization. So in order to study XALD, we started characterizing different ABCD1 mutant zebrafish with either a premature stop codon or a splicing defect. And these fish happen to have a decreased expression of ABCD1, shown on the left by RT-PCR. And more importantly, the fish recapitulate the major biomarker of the major biochemical abnormality in ALD, which is the accumulation of very long-chain fatty acids. The expression of the genes encoding the major myelin proteins in zebrafish, like PLP1A, MBP, and MPZ, was measured by RT-qPCR. And we observed a decreased expression in PLP1A, suggesting a defect in oligodendrocyte function. And this is correlated with observation by electron microscopy, which display decreased myelination of axons in the ventral spinal cord. So finally, to test the motor function, we tracked the swimming behavior with this automated setup in a 96-well plate format that I presented earlier, and we record the fish larvae movement for about 5 minutes after light stimulus. And by measuring this light-evoked response, we also observed a decreased swimming function in ABCD1 mutant zebrafish. So just to summarize briefly that first part, we've seen that similar to patients with ALD, zebrafish ABCD1 mutants have elevated VLCFA levels, and the CNS development is disrupted with hypomyelination in the spinal cord, abnormal patterning and decreased number of oligodendrocytes, and also increased cell death. And after 5 days, ABCD1 mutants already demonstrate impaired motor function. So these results have been published a few years ago in Human Molecular Genetics. I encourage you to read the publication if you are interested. So obviously, after noticing the phenotype, the motor phenotype of this fish, we had quickly in mind that it would be a great model to start a large high throughput drug screen. And so we use the paradigm that I presented earlier by treating the fish for about 3 days with different compounds and measuring their swimming behavior at 7 days with this light evoc response, recording for 5 minutes their behavior. And so we tested a drug library of about 2,500 compounds and measuring 5 different variables based on this behavior recording, and we selected the more promising compound for a rescreen. And after this rescreen, we happened to identify chloroquine and prednisolone, which happen to be the most promising candidates. And here, looking at the Z-score in red, we can see that chloroquine at the dose of 10 micromolar is actually seems to be the more promising candidate. And so on the bottom, we can see just similar results as a dot plot. We can see in 2 different ABCD1 mutants that the swimming behavior, which is significantly decreased in the mutants, is rescued after treatment with chloroquine. And so when we look in the spinal cord of these mutants, we can see a decreased number of oligodendrocytes at 7 days in ABCD1 mutants, and after treatment with chloroquine, we see this decreased number of oligodendrocytes being rescued. So to investigate the mechanism of action of chloroquine, we tested the expression of different genes involved in very long chain fatty acid beta oxidation, or also in myelination, and we observed this significant increase in this gene SCD, which appeared to be quite interesting. And so one of the reasons it was very interesting for us is that pretty much at the same time in the group of Stefan Kemp, they realized a pretty similar screening with patient fibroblasts. And they also happened to identify chloroquine as one of their hits. And here, when they measured the synthesis of saturated very long chain fatty acids, you can see that the very long chain synthesis increasing in ALD patient, in ALD fibroblasts is significantly decreased in fibroblasts treated with chloroquine. And interestingly, when they treated these fibroblasts also with an inhibitor of this enzyme SCD, we can see that the rescue with chloroquine is to a lesser degree, suggesting that at least partially the mechanism of action of chloroquine is through the induction of this gene SCD. And so what the enzyme is actually doing is it's catalyzing the desaturation of long-chain fatty acids to monounsaturated long-chain fatty acids. And these are basically what are fueling the elongation and generated those very long-chain fatty acids. And so instead of, in ABCD1 deficient cells, instead of accumulating saturated very long chain fatty acids, the cells start accumulating monounsaturated ones. And one of the reasons it's very important is this nice experiment that was realized also by the Stefan Kemp group. They also measured the effect of saturated and monounsaturated VLCFA toxicity by looking here at XBP1 by Western blot, which is a marker of ER stress, as also controlled by tunicamycin. And they measured that, they observed that saturated VLCFA were actually more toxic in ALD cells, that saturated VLCFA, sorry, were actually more toxic to ALD cells compared to monounsaturated ones. And so it's suggesting that the induction of the expression of SCD1 would be a nice strategy to protect the cells from the toxicity of saturated VLCFA by switching them to non-toxic monounsaturated VLCFA. And so they also realized this very nice lipidomic study measuring the different complex lipids that were accumulating in ALD cells, here in red, And you can see that after treatment with an LXR agonist, which is also inducing the expression of SCD1, that a good number of those complex lipids were actually rescued. So again, chloroquine was identified as the top hit of the ALD zebrafish motor behavior screen. Chloroquine induces the expression of SCD in ALD zebrafish, which is the autolog for SCD1 in humans, and which is the enzyme regulating the balance between saturated and monounsaturated VLCFA. So just a quick more insight on the mechanism of action of chloroquine, which is this lysosomotropic compound known to affect the pH in endosomes, and is also often used experimentally as an inhibitor of autophagy. And so with that in mind, we also tested a similar compound, lysosomotropic compound, which is called bafilomycin, which is also an autophagy blocker. And you can see on the right that it doesn't have the same effect as chloroquine, and it's not able to rescue the motor phenotype in ABCD1 mutants, which is really suggesting that there's a distinct mechanism of action of chloroquine, which is at least partially through the induced expression of SCD. And that's something that we're really interested in and want to investigate. And so similar to chloroquine, also LXR agonists can increase the expression of SCD and can normalize the saturated very long-chain fatty acid levels. And so shifting saturated VLCFA towards unsaturated VLCFA can protect against ER stress. Now to extend the discussion a little bit, I want to mention the evidence suggesting a role for ER stress in the ALD pathology with work from the CAM group and also the Puyol group, respectively indicating that VLCFA are leading to cell death through this pathway, and that the enfolded protein response, which is the major component of the ER stress, is well activated in a mouse model of ALD, and that it represents a good drug target. And I also want to mention that amongst the genes that are regulated by this pathway, many genes are involved in lipid biosynthesis, and amongst this gene, SCD1 is one of them. And now that we've seen the role of SCD in ALD, it's interesting to note the existence of this sort of feedback loop. And keeping in mind that ALD is a complex disease with various factors potentially responsible for the very distinct manifestation of the disease, so we wanted to generate a two-hit model where a second factor could be precipitating the pathology. And with this evidence for ER stress being a major compound, tunicamycin, which is commonly used experimentally for ER stress promoter, it just appeared to be a good candidate. So I treated the ABCD1 mutant fish or their wild-type siblings with tunicamycin for 3 days before measuring the expression of ER stress effectors like ATF4 or CHOP, which are inducing apoptosis in the cells. And we can see that their expression is significantly increased in ABCD1 mutants after the treatment, which is suggesting that ABCD1 mutants are more prone to ER stress after the treatment with tunicamycin. And also with these two-hit models, we had the idea of combining the ER stress with also a drug promoting inflammation, which is this toll-like receptor agonist lipopolysaccharide. And quite surprisingly, instead of worsening the phenotype, which is what we were expecting, we can see that the LPS preconditioning happens to improve both the survival and the swimming function, which is decreased in the fish, ABCD1 mutant fish after treatment with tunicamycin. And with LPS, it's actually rescuing a little bit the phenotype. And apparently it's something that has been studied in different models. And the preconditioning with LPS happens to protect against apoptosis induced by the ATF4 CHOP pathway. And on the other hand, we looked at the activation of XBP1, and we can see on the right that mutants that were treated with tunicamycin and with LPS showed no difference in the XBP1 splicing. And so this is really suggesting that the TLR and ER stress signaling have a common effector, and they could potentially be involved in the neuroinflammation. So finally, I also investigated the effect of tunicamycin on the innate immune response in the brain in ABCD1 mutant fish, and I used this living dye called natural red, which stains for microglial cells and infiltrating macrophages. And we observe this very important redistribution here on the left of immune cells, which seems to be surrounding the major arteries in the fish brain. And these preliminary results, I'm not really sure how to explain the decreased number of myeloid cells in ABCD1 mutants, but we can see with tunicamycin the significant increase in the number of cells infiltrating the brain of the fish. And so this seems to be something suggesting that there is mechanisms resembling neuroinflammation in this model. Unfortunately, I was hoping I could present some transcriptomic and lipidomic results here as we have this analyzing going on. And we expect to measure the effect of tunicamycin with these omic techniques, and in parallel also controlling what chloroquine is actually doing in this fish. We also want to investigate the events leading to the blood-brain barrier disruptions and what could produce this innate immune response after the treatment. And finally, it will be also interesting to compare and to control the effect of SCD1 inducer and especially chloroquine on this model. So to conclude and summarize, our Zebrafish model of ALD recapitulates key features of human disease pathology and reveals novel features of underlying disease pathogenesis. In the second GCI paper, we showed that metabolic rerouting of saturated to monounsaturated VLCFA may alleviate the lipid toxicity, and this strategy may be beneficial in ALD and other peroxisomal disease in which VLCFA play a key role. And our current work focuses on a novel 2-hit model with characterized ER stress and CNS inflammation, which is suggesting a promising model to study the physiopathology of cerebral ALD. So these are the references and publication I used for the presentation, and with this I want to thank you for your attention. I want to thank all the collaborators from the Josh Bunkowski lab, the Zebrafish facility, and also the collaborators from the University of Amsterdam and Vienna, and also the European Leukodystrophy Association, which has been funding part of this study. Thank you.
Thank you. Do we have any questions? And I'm sorry, we also have people that are asking questions online.
Sure.
We might have skipped them from the last speaker, but if there are any questions online, please let me know. Oh, there's a question in the audience. Go ahead, Patty.
Amazing work. I have to say from being in the bench, and for those that don't do research, there are thousands of hours of work, and previous to your work, people that made it happen. So congratulations on the work that you're doing on generating an animal model that also has features of the disease that allows us, as you very well pointed, to test different mechanisms and drugs and things. I had a question about the neuroinflammation. When you show that picture of the fish head, the cells, I thought those were the venous sinuses. That's the venous system, not the arterial system. Uh-huh. They're trying to probably either get out or sensing something in the venous system. Do you also see them deeper in the brain? That the number of cells that you quantify, how Because that looked very perisinus, perivenous stuff.
Yes, that's a very good point. Yes, so it's most likely at the end of the optic tectum. So it's mostly the peripheral cortex, and not so much deeper into the brain. We cannot see the same cells now. That really seems to be closer to the meninges, I guess. I don't know if that.
Does the spinal cord also have?
The spinal cord do have a little bit of cells, but we don't see any effect of tunicamycin in the spinal cord. So it's really differential, yes. Or at least it's a lot easier to observe in the brain than in the spinal cord.
We have someone on Zoom asking if chloroquine can diminish the VLCFA levels in rich VLCFA conditions. They're wondering because they think that chloroquine can lower VLCFA accumulation at early stages.
I'm not sure I got the second part, sorry, of the question. Could you repeat that?
The second part was they said, I think that chloroquine can lower VLCFA accumulation at the early stage. So they're wondering, can it diminish VLCFA in rich VLCFA conditions?
That's a very good question too. So we don't really have a great model with zebrafish to enrich for VLCFA, as we can only measure the normal VLCFA conditions. And so I cannot really tell for that. Maybe Stefan Kemp can bring
I'm happy to take over, Quentin. I think it's I totally understand the background of this question, and I think it's really important here to mention that the effect of chloroquine on variant chain fatty acids is lowering it, but it's not normalizing it. So if this is a question coming out, what I understand, and I think the real question behind it is, should I take chloroquine? No. Because a lot of the data that Quentin showed was with another compound, and that compound is unfortunately toxic to humans, and that really normalizes the fatty acid. So the lipidomics that you showed was with the TO903017 compound, and so in the beginning you showed chloroquine, but it lowers it like 20%, but only in a cell model and in the zebrafish. So no, do not take chloroquine. It doesn't help against COVID. It doesn't help against ALD.
So yes, that's a very good point. There's a toxicity associated with both compounds, either the LXR agonist, and that's also the case for chloroquine, especially considering ALD when we want a chronic treatment. So something to keep in mind, yes.
I myself had a question. Do you, given the early onset presentation in the zebrafish, how is are there ABCD1 homologs in the zebrafish the same as in humans? So how is ABCD2 expression and so on? Why is it that the zebrafish exhibits oligodendroglial abnormalities that early as opposed to mice or patients?
That's a very good point. Very good question. So zebrafish do have the same ABCD transporters, peroxisomal transporters. So there is an ABCD2. I don't know if the expression is similarly that what is in human mirrored kind of in with a tissue-specific expression. So it's likely that we can see the gene expressed very early in the brain development. So it's likely to be an important peroxisomal genes early in development, and I'm guessing that's what could explain a difference. Maybe we don't see ABCD2 expressed as early, and no, I don't really have any good observation to explain that.
More questions? No more online questions? All right. Thank you very much. Thanks. That was a great talk.